right, so I'm happy to talk to you a little bit about some of our work in nutritional biomarkers today. And before I begin, I will say that I'm a nutritional epidemiologist. I'm also a cancer epidemiologist, and a lot of our work looks at nutrition and cancer etiology. A lot of it works at other risk factors in cancer etiology, but today I'm only going to talk about one aspect, which is nutritional biomarker work, as Dr. Lynch alluded to. I have nothing to disclose. All right, by way of introduction, I think everyone in this room recognizes that diet is an important cause of human cancer. But just to give you some feeling for what the current estimates indicate, is that diet and lack of physical activity together account for roughly a third of all cancers in the United States. However, despite that recognition, it's hard to know the exact role that diet and various dietary constituents play in human cancer. And that's because there's all this inconsistent data in the literature. I mean, one week a study comes out, says fiber is protective. Next week a study comes out, said fiber is not related to human cancer risk. So we have all these inconsistencies in the literature. And what's recently been recognized is that diet is really hard to measure. There's a lot of measurement error with regard to dietary estimates. That measurement error impedes research, and that contributes to a lot of the inconsistencies in the literature on nutrition and cancer. So to kind of to highlight this a little bit more clearly, in terms of difficulties in accurately reporting dietary intake data, currently we have biomarkers in the field that are what we call reference biomarkers. And one reference biomarker is called doubly labeled water, and that's an accepted reference biomarker for energy intake. And a landmark study that was completed within about the past 7 years or so used this biomarker in hundreds and hundreds of individuals and asked the question, is how valid is self-reported dietary intake as compared to this reference biomarker? And what I'm going to show here are just the estimates from the males of the study. There were several hundred males that were given doubly labeled water. They were asked to report diet by 2 different methods, and this is what the findings basically showed. So using the doubly labeled water biomarker, the estimate was that these men actually consumed about 2,800 kcals per day. When the men reported using a 24-hour recall, they reported approximately 2,500 kcals per day. And the recall is not that commonly used in nutritional epidemiology work. The primary dietary instrument that has been used in nutritional epidemiology is this food frequency questionnaire here. And based upon that methodology, the men reported 1,900 kcals per day. So we see an awful lot of underreporting, and that's an issue. We all know it happens. We know it's a problem. But what's an even greater problem is it's not consistent. So it varies by sex, by age, and especially by BMI. So in the same study, the normal weight males, 22% of them underreported energy by 30% or more. In contrast, in the obese males, 53% of them underreported energy by 30% or more. So not only do we have underreporting, but it's not consistent across different groups. And so this is a real problem in terms of nutrition and cancer research. So in response, the NIH put out a program announcement, and they said we have got to do better in terms of improving the assessment of diet and physical activity. And they noted that there are new, more innovative methods that are really needed, that we need to bring in innovation into the field of nutrition and physical activity research. So this brings me to the carotenoids. The carotenoids are something that I've worked on for the past almost 3 decades now. Carotenoids are plant pigments, and they are responsible for red, orange, and yellow colors in nature. They protect plants against photooxidative damage. They are widespread in different fruits and vegetables. The primary dietary carotenoids include beta-carotene, which is the main reason oranges are orange. Alpha-carotene, also found in carrots. Carrots are orange. Alpha-carotene, also found in carrots. Lycopene is a red carotenoid that's responsible for the red in watermelon and in tomatoes. Lutein is one that's found in many different foods, including corn. And beta-cryptozanthin is the reason that oranges are orange. So humans ingest all of these carotenoids. We absorb these carotenoids into our blood and into our tissues. And carotenoid concentrations in humans have traditionally been considered the best biomarker of fruit and vegetable intake. And you see I put spinach here. Spinach also contains a lot of carotenoids, but the color is masked by chlorophyll. So green leafy vegetables are very rich sources of dietary carotenoids as well. Now, traditionally, people have measured carotenoid status in human studies using blood samples that have been subjected to HPLC analyses. So you get a chromatogram, usually reverse phase HPLC. It's about a 20-minute chromatographic run to measure carotenoids in blood. And that has been the traditional carotenoid biomarker that's been used for human research. It's got a lot of advantages. It's widely used, and you can get individual carotenoid values. But there are some key disadvantages as well. It's invasive. You have to collect blood samples. It's expensive to process, prepare all these samples, run them through the chromatographic systems. And carotenoids have a relatively short half-life in blood. Approximately 2 weeks is the half-life of carotenoids. So the values fluctuate in response to recent meals. Nonetheless, despite this limitation, carotenoid concentrations are inversely associated with risk of many health outcomes. That is, people who have higher blood levels of carotenoids have lower risk of cancer and cardiovascular disease and other health outcomes. And just to give you a sense for what this looks like, this is an example of a plot from a study which was a cohort study looking at all-cause mortality as a function of plasma carotenoids. And you can see that people who have high plasma concentrations of carotenoids are more likely to survive over a longitudinal follow-up period as compared to people who have low plasma concentrations of carotenoids. So carotenoids are a biomarker of better health status. They are derived from dietary intake of fruits and vegetables. So arguably, carotenoid status could someday become routine because it predicts health outcomes, just like serum cholesterol does. But in order for that to happen, we're going to need rapid, inexpensive assessment tools for carotenoid status. And so some years ago, our research team proposed a new method for non-invasive assessment of carotenoid status using resonance Raman spectroscopy. And essentially what we're trying to take advantage of is the fact that carotenoids concentrate in skin. So for example, if any of you know vegetarians or other people who eat a lot of vegetables, the palms tend to get kind of yellow-orange. That's due to the accumulation of carotenoids in the skin. Others, babies, you see those babies that have those bright orange cheeks. They're eating pureed pumpkin and squash and spinach and all these pureed vegetables. Those carotenoids are accumulating in the skin, and you can visually see that. And we're trying to take advantage of that to measure carotenoid status non-invasively. So what is resonance Raman spectroscopy, and how do we propose to use it? It's basically using a small probe with a laser to deliver light to a tissue of interest, in our case, human skin. And the Raman spectra reflects the vibrational, rotational energy of molecules in the skin. Carotenoids are particularly strong Raman scatterers when they are radiated with light at 488 nanometers, and that's close to the lambda max for carotenoids. And lycopene, in contrast to the others, has greater scattering at a higher wavelength, 514 nanometers. 488, this is blue light, and 514 is green light. And so presumably you could take advantage of these characteristics to measure carotenoids in skin. Now the reason carotenoids work so well for Raman scattering is based upon their structure. So this is beta-carotene, this is lycopene, they're both C40 compounds. What they have in common is this long series of conjugated double bonds. They both have 11 conjugated double bonds. So that vibration and stretch in response to a particular wavelength of light is a unique feature of carotenoids that makes them suitable for Raman resonance spectroscopy. So before we could begin to use this tool in epidemiologic work or in human clinical studies, anytime you start to develop a new biomarker, you have to understand something about how that biomarker varies within a person and how that biomarker varies across people. So these are some of the things you'd need to understand about this biomarker. You also need to know if it's accurate and if it's valid as compared to some gold standard. So we wrote a methodology grant to the National Cancer Institute to try to develop this biomarker for human subjects. And it was funded by the NCI as an R01 methodology development grant. The aims of our grant are as follows. The first aim of this proposal was to basically build the instrumentation, including trying to separate lycopene from the other carotenoids. And this work was done by our partners in the Department of Physics at the University of Utah. So we partnered with some laser physicists to develop the instrumentation that we needed to develop this biomarker. And what they built for us was our first Raman scanner, and this was our version 1 Raman scanner. This is the entire Raman instrument. It includes 2 lasers, one at 488 nanometers and one at 514 nanometers. Includes all the CCD detector, the camera, everything for the data collection, and it's interfaced to a laptop computer. The Raman light is produced through this fiber optic tube here, and this is the probe that can be placed into contact with the skin. And you can see there are 2 wavelengths. This is the blue one, and this is the green one. This measures total carotenoids. This measures lycopene. And as I'll show you a close-up, the laptop is actually where the interface is for the data collection. So that was our first generation Raman scanner. This is showing how you would use this in a clinical setting. These are 2 research assistants. Dr. Brenda Cartmell is here, and one of our graduate students just demonstrating how this would be used. In this case, you'd be measuring it in the inner forearm. And we tried many different body sites, as I'll show you in a moment. The laser that we're using is a class 2B laser, and so it's analogous to a laser pointer. And so, you're not supposed to put laser pointers in people's eyes. Same thing with this. We basically have to wear protective eyewear in order to meet laser safety standards. So in all of our research settings, you will see that we're wearing protective eyewear because it is analogous to a class 2B laser, like a laser pointer.
And everything looks kind of orange that way, too.
Good point. All right, this is what the data collection interface looks like in this study. So it's like area under a curve. This is a processed Raman spectra. This is the blue wavelength for total carotenoids. This is the green wavelength, which gives us lycopene. And 2 things to note. We actually get a numeric value. That's our total carotenoid Raman intensity score from the software, the processing software. The green is our lycopene measure there, which we get there in the second one. And then the other thing I wanted to point out is this is a 30-second scan. So in a 30-second scan of human skin, we're able to estimate the dermal carotenoid concentration. So the second aim was, now that we had this instrument, to get it into people and to try to assess interperson variability over time and across different body sites. And we decided to do this over a 6-month window of time because that would maximize the seasonal variation we would expect to see in fruit and vegetable intake. And as you can see, we used 3 body sites: the palm, the inner arm, and the outer arm in this particular human study. So for this study, we recruited 75 people. We asked them to come in for 6 different scans, one at baseline, at 1 week, 2 weeks, 1 month, 3 months, and 6 months. And the goal was to examine both the distribution, like how variable are these carotenoid levels in this population, so the distribution and the reproducibility of dermal carotenoid Raman counts in this first human study. So what's shown here is the distribution. These are the baseline measures for our 75 study subjects in this first human study. And we were actually really pleased when we got these results because it looks nicely normally distributed. We have some people who are really high, we have some people who are really low, and we have a group that's concentrated here in the middle. So that was actually really attractive to us. And in part, I think part of the reason it's normally distributed is the estimated half-life of carotenoids in skin is long. It's like 3 months or longer. So in blood, you get real skewed distributions because it's got such a short half-life. If somebody has a high carotenoid meal, you get these long tails. We're not seeing that in skin. So we were pleased with the distribution. The next thing is the reproducibility. And I wanted to give you a visual graph of what the reproducibility looks like. These are 4 consecutively enrolled study subjects, number 25, 26, 27, 28. And these are their 6 times that we scanned them. And what you can see is that the numbers bounce around a little bit. However, the people maintain their relative rankings quite consistently. So for example, this person here was one of the highest in total carotenoids at baseline and remained high throughout this particular 6-month window. If you compare that to somebody like this person down here, this person had the lowest lycopene measures of these 4 study subjects and stayed low throughout, even though there was a little bit of movement around this biomarker. So it's reasonably reproducible. The way we quantify that is with the interclass correlation coefficient, and the interclass correlation coefficients within a body site are shown here. So for example, in the palm, the ICC is 0.97. 1.0 would be considered perfect. So it's very high, namely that subjects who have high dermal carotenoid status in the palm at one time point are high at the other time points. This is the inner arm, and this is the outer arm. So our ICCs were best in the palm, but they're high in all 3 body sites. So what this is telling us is for human research, this is a highly reproducible biomarker reflecting kind of their usual status of carotenoids. Next is the ICCs across the body sites. And the reason this is important is if we're going to be measuring the palm, we want to know, is it reflective of what's happening in other skin sites? And so it's asking the question, if someone is high in the palm, are they also high in the inner arm and the outer arm? And is it a marker of systemic status? In this case, you can again see the ICCs are very high, approaching 0.9, suggesting that it's a reasonably good biomarker of overall status when you measure one body site. So the next aim of the grant was to look at the validity of this measure, and that is, you know, hypothetically, this should be measuring carotenoids through Raman resonance spectroscopy, but the only way to verify that it's truly working was to actually do dermal biopsies and assess dermal carotenoids by HPLC in dermal biopsies. So for this part of the research, we partnered with David LaFell's group from the Derm Surgery Suites here at Yale, and we enrolled study subjects who had to agree to come in. They had to be scanned with our Raman scanner, and then they had to undergo a dermal biopsy, a 3-millimeter punch biopsy, that we then extracted the carotenoids and did HPLC analysis. And that would be the test of whether or not this is performing, is measuring actually the carotenoids in comparison to an HPLC methodology. So here's the results from our biopsy study. This is looking at total carotenoids in skin by HPLC, and total carotenoids in skin based upon our 30-second RRS scan. The correlation coefficient is 0.66 and highly statistically significant. We would have loved to have had a higher correlation, 0.7, 0.8, but recognize we're working with very teeny amounts of skin. We're working at really low levels of carotenoids, and there's errors in the HPLC method as much as there are potentially with the Raman scan. So nonetheless, it's reassuring that what we're capturing is in fact dermal carotenoids. And this is the data for lycopene. Slightly higher correlation, 0.74, highly statistically significant. It may be slightly better because here we're only looking at one carotenoid, not several different carotenoids that all have slightly different spectra. So it looks like it's measuring essentially what we want it to be measuring. So we were encouraged by these findings as well. We also looked at the correlation with blood carotenoids, and that was highly statistically significant. And we also looked at the correlation with self-reported fruit and vegetable intake, which was also correlated, statistically significant, not as highly correlated as you'd expect because dietary measures of carotenoids are rather crude. But nonetheless, it's reflecting blood, it's reflecting diet. So we were then encouraged about moving this into larger population studies, but the device that we had really had limited portability. So we went back to our laser physicist partners and said, can you build us a more robust and portable scanner for our use? And they said, yes, we can, but we can only use one wavelength of light. That would be blue light to assess total carotenoids. So they built for our use a 2nd-generation Raman scanner, which is shown here. And you can see it's about the size of a shoebox, and it's interfaced with a laptop computer. And it's robust and it's portable, so we can pack this up and take it into a field setting in order to measure human subjects for clinical and epidemiologic research studies. So this is showing, these are 2 of the research assistants showing how you would presumably measure this, you know, in palm. In this example here, the probe is interfaced with the palm. And this led us into a study in children. And you may say, well, why is a cancer epidemiologist focusing on studies in kids? Well, the reason is that early life nutrition is of huge interest for people who are interested in chronic disease epidemiology. Moreover, it's really hard to assess diet in kids. It's really hard. Parents don't know what kids are eating. Teachers don't know what kids are eating. Nobody knows what kids are eating. So we said, well, this might be an ideal setting to use a rapid, non-invasive measure of nutritional status for research purposes. So we went into a population of preschool children. And we had the opportunity to partner with Valerie Duffy from the University of Connecticut, who was doing nutrition studies in preschool kids in Waterbury and Meriden, Connecticut. So we had access to a population of almost 400 preschool kids that we could try this technology out. So you can see the kids are wearing their miniature-sized protective eyewear. They're smiling, they're having a good time, and they're getting their dermal carotenoids assessed for this study. So, to make story short, this is the values we found in our preschool kids. Again, a nice normal distribution. First time we tried it in kids. These were all low-income preschool kids that were in like Head Start programs. But we had some kids who had really high status, some kids who had really low status, and then a group clustered here in the middle. When we looked at the predictors of carotenoid status in these children, what we found is those kids whose parents had reported that they liked fruits and vegetables and high carotenoid foods, they had higher dermal carotenoid status. Also, we had a crude measure of fruit and vegetable intake and total carotenoids by a parent-completed food frequency questionnaire. That predicted higher dermal carotenoid status. And age, we had a range from 3 to 5. The older kids had higher dermal carotenoid status. And I don't know if it's because they're eating more fruits and vegetables or they're accumulating more as they get older, we're not sure. But that predicted positive status. In contrast, negative predictors were shown here. The first one is we had demographic data on these families, and the families who reported participating in the food stamp program, those children had significant and substantially lower carotenoid status. So whatever poverty-induced deficiencies food stamps is designed to overcome is not happening in these kids. They had significantly lower dermal carotenoid status in our research. And BMI, the heavier the kids, heavier overweight and obese kids had lower dermal carotenoid status in our study as well, which is consistent with what we know about our human adult data, as well as blood data, that obesity predicts lower carotenoid status. So in summary, to make this a really short story, we think that this is a promising objective indicator of fruit and vegetable intake and carotenoid status in both adults and in kids. We had low within subject variation, high between subject variation, which is what you're looking for with a biomarker. We had significant correlation with carotenoids measured in tissue, and we're the only ones to have validated this technology using tissue biopsies. And we therefore conclude it's a promising biomarker for epidemiological and clinical research. So last, I just need to acknowledge this was a huge team effort. A lot of people worked on this project. Brenda Cartmill and Stephanie Scarmo. Stephanie was a graduate student who did some of this work for her dissertation in the School of Public Health. These are some of the students here who assisted with the data collection. These are our physics partners from the University of Utah. These are the people who did some of the biochemistry work with the skin biopsy samples. David LaFell and his group allowed us to do these dermal biopsies. We had a biostatistician. UConn people helped us do the preschool work. And lastly, the NCI who funded this work. So it looks like I have about 3 minutes left for questions.